In the cytoplasm, one molecule of glucose is broken into two pyruvate molecules. This process is anaerobic, meaning it doesn't require oxygen. In the energy investment phase, the cell uses 2 ATP. Then, in the energy payoff phase, it produces 4 ATP and 2 NADH, leaving a net gain of 2 ATP. Then, each pyruvate moves into the mitochondrial matrix, where it combines with coenzyme A to form acetyl-CoA. During this step, one molecule of CO2 is released per pyruvate and reduces NAD+ to NADH. Some FADH and FADH2 will appear later to help move electrons in the next stages. Now we enter the citric acid cycle, also known as the Krebs cycle inside the mitochondrial matrix. This is where aerobic respiration occurs, meaning that oxygen is required. Acetyl-CoA joins with oxaloacetate to form a 6-carbon molecule. Through a series of redox reactions, we make 3 NADH, 1 FADH2, 1 ATP, and release 2 CO2 per turn. Water helps to keep things running smoothly. At the end, oxaloacetate is regenerated to start again. The NADH and FADH2 may now donate their electrons to the electron transport chain on the inner mitochondrial membrane. As electrons pass through protein complexes, H ions are pumped into the inner membrane space. Then those ions flow back through ATP synthase, powering oxidative phosphorylation, where the majority of ATP is made. At the end, oxygen accepts the electrons and forms water. This is a drawing that I made to represent cellular respiration. Light-dependent reactions occur in the chloroplast, specifically in the thylakoid membranes that are stacked into structures called grana. The process begins when sunlight strikes chlorophyll in the photosystems. The light energy excites electrons, which travel through an electron transport chain. This creates a proton gradient. Protons flow through ATP synthase to make ATP by chemiosmosis. At the same time, electrons reduce NADP+ to NADPH. ATP and NADPH head to the stroma for the next phase. This is a drawing to represent the light-dependent reactions. In the stroma of the chloroplast, the Calvin cycle uses ATP and NADPH from the light-dependent reactions to convert CO2 into glucose through 3 main steps: carbon fixation, carbon reduction, and carbon regeneration. First is carbon fixation. CO2 attaches to another molecule with the help of Rubisco to form a 3-carbon compound. Next, carbon reduction. ATP and NADPH provide energy and electrons to reduce the carbon compounds into G3P, a high-energy sugar. Some G3P molecules leave the cycle to help form glucose, while the rest are used in carbon regeneration. Finally, carbon regeneration. This process rebuilds the starting molecule, allowing the cycle to continue as energy ATP and NADPH are used. They turn into ADP and NADP+, and they are ready to return to the light-dependent reactions. Through these 2 stages, photosynthesis transforms light energy into chemical energy, producing glucose to fuel the plant and oxygen for the environment.